Well, hello everybody. Today we're going to be talking about our human DNA testing lab, which is also called the taster lab. Now, you guys didn't get to do this, so we're going to be going through the experiment and kind of pretending that we did it. And what we're going to do is analyze the data from previous experiments that other students have done. So it's not quite as good as doing the same thing, but it's pretty educational and informative anyway. Okay, so we're going to do human DNA testing, and it's pretty standard how this all starts out, regardless of whether you're doing it in a lab or shipping your DNA off to a testing service. You need to collect a cell sample from people. Saliva and cheek cells are the most widely used. Places like 23andMe use saliva. In our lab, we use cheek cells. So you can sort of see why we couldn't do it this lab. You can't be flinging around clinical samples in the middle of a pandemic. So this was a definite one that we really were not able to do, unfortunately. Anyway, once we get our cell sample, we need to extract the DNA from the cells and then amplify selected portions using PCR, which we'll explain in a little bit longer. Then we will analyze the PCR amplified DNA in some way. And it's this analysis of the PCR amplified DNA that's going to enable us to distinguish one allele of a gene from another. So what we are looking at, what the gene we're looking at, is called TAS2R38, cumbersome name, I know. It's a gene that encodes a receptor that's in your mouth that's responsible for tasting or your ability to taste certain bitter substances. Okay, so there are DNA sequence variations in this gene, and this was actually discovered totally by accident, that people with some sequence variations are able to taste this particular compound called PTC, and to them it tastes very bitter. Other people with slight variants in their protein sequence don't taste it at all, and to them it tastes like paper. It tastes like absolutely nothing. So the big T is the taster phenotype, which is dominant to the little t non-taster allele. And so in our class, we gave people the PTC paper, and they could tell us what their phenotype was, which is to say whether they could taste the paper or not taste the paper. Okay, so in terms of how we're going to actually do this and looking at what we're doing, there are actually three amino acids that differ between the big T taster allele and the little t non-taster allele. And we're going to be testing for those single nucleotide differences that occur at 2 out of the 3 positions in the protein. So one position is a G to C, another one is C to a T, and the other is a G to an A. But we're only going to be looking at 2 of those. So first we isolate the cells. We use protease K, Chelax, and heat to break them open and to extract our DNA. Okay, one thing to think about, like why do we need PCR anyway? We've got plenty of DNA in our cells. Well, that's certainly true, but we're only going to be looking for 2 particular nucleotides in this gigantic haystack of a genome that we have. Each cell has approximately 6 times 10 to the 9th base pairs of DNA, and we are looking for 2 of those nucleotides. So that's why we have PCR to help us out with this. So if you just isolate human DNA and just run it on a gel, it looks like a huge smear. You can't distinguish individual molecules or individual genes, even though they are present. We need to apply the PCR technique to selectively synthesize or amplify the region of interest that we want to look at so that we can analyze it further to look for what alleles we want. Okay, so what PCR does, of course, is selectively synthesize particular regions, and we're calling this the target sequence. So in our case, the target sequences are specific parts of the TAS-R238 gene, and those regions that we're amplifying, our region of interest, are going to contain the nucleotides that we're looking for. Okay, so we're looking at a C versus G at one place and a C versus T at another place. So we obviously need many more copies of the DNA that we can use then to test which of those alleles is present. Okay, so visualizing what this looks like, I'm showing the T allele with the actual 2 strands, and there's a C to G at one position. The little t is a G to C at that position. And the other position we're going to look at is a CG base pair, which is modified to a TA base pair in the little t allele. So those are the single nucleotide polymorphisms that we're looking for. Single nucleotide polymorphism just refers to a position in the genome where people differ in one particular nucleotide pair. Okay, so we use PCR to selectively synthesize DNA from our regions of interest. So remember, we had the big T allele and the little t allele, and I'm just diagramming what the PCR is going to look like. Now, everybody's DNA is going to amplify in exactly the same way with this particular pair of PCR reactions, because all we're trying to do is make enough DNA surrounding the nucleotide of interest that we can then test the PCR product for what actual nucleotides are present. So the one set we're using is going to give us a PCR product of 221 base pairs. The primers are indicated by little arrows pointing into the region to be amplified. The other PCR reaction we're going to do looks for this GC versus AT. And again, we can't just synthesize that. We synthesize a region surrounding it, which is going to give us a product of 303 base pairs. Again, the primers are shown with little arrows indicating where the amplification actually starts for those two reactions. Okay, so the slide above indicates or suggests that we'll have amplified two different regions of the TAS gene. We will create two different PCR products, one for each of the polymorphisms that we're looking for. So important to realize everybody will have these same products, irrespective of what actual CG or whatever allele they have. But now we need to find out which alleles we have. And that's going to rely on a little bit convenient trick of these particular nucleotides. Okay, so we want to know what alleles we have. Well, one thing we could do is we could just sequence them, right? This is certainly possible, but that's not what we're doing. We're going to use a very convenient trick, which is certainly just a coincidence, really, that the single nucleotide differences between the taster and the non-taster alleles actually alter the presence of particular restriction sites. So we're going to look for those restriction sites based on just cutting with the enzyme and running it on a gel. And that's what's going to tell us, based on the sizes of the products, which alleles are present, which ones cut and which ones do not cut. Okay, so here's the first one. This is the restriction enzyme FNUH1. Its cut and recognition sequence is written GC, N, GC, where N is anything. So you need to have that sequence perfect or else the enzyme will not cut the molecule. So the taster allele actually contains a site right there. This is a GCTGC, which conforms perfectly to the enzyme recognition and cut sequence. On the other hand, the second allele, the non-taster allele, does not cut because that CG base pair of the taster allele is a TA base pair in the non-taster allele. And that destroys the restriction site because it's not a GC anymore, it's a GT, and that will not cut, the enzyme will not cut at that particular allele. So we're going to be able to distinguish the person's big T versus little t genotype based on partly the ability of this enzyme to cut that particular PCR product. Okay, so what this looks like is we have this 303 base pair PCR product, and we're going to treat it with the enzyme FNU4H1, and the big T allele has the recognition sequence as shown, and so I'm indicating the fact that it cuts with an arrow. It's going to cut on both strands, obviously. And so what's going to happen if you got a big T allele is that 303 base pairs is going to get broken up into two pieces, one 238 base pairs and the other 65 base pairs. That's for the big T. The little t allele does not cut because it has a GT instead of a C, and so you can treat it with the enzyme all you want. It will remain to be 303 base pairs, irrespective of whether you try to treat it with the enzyme. And these differences can be seen on agarose gels. Okay, so we're going to get different results depending on whether the person has a big T, little t, or little t genotype. Remember, we're diploids. We've got two alleles of each gene. And you could have two taster alleles, one taster and one non-taster, or two non-taster. Now, the big T is dominant So the big T and the big T little t people will be able to taste the PTC, whereas the little t cannot. So remember the sizes here, that's going to be important later. The big T produces a 238 base pair piece and a 65 base pair piece. The non-taster does not cut with the enzyme, so the PCR product remains full length even when the enzyme is present. And these are differences that you can see by agarose gel electrophoresis. Okay, so summary for the FNU enzyme. The non-taster allele is 303 base pair, and the taster allele cuts 238 and 65. Okay, I'm just going to describe the HAE3, which is the second site. That full-length product is 221 base pairs, and if you are a taster allele, you will cut that into two pieces, 177 and 44 base pairs. Same idea as the FNU. The HAE3 enzyme site is destroyed by the nucleotide change that occurs with a non-taster. Okay, so here's a picture of a glass gel. We ran the gel, regular old agarose gel. These are bigger gels than the ones you have, but the idea is exactly the same. So to use these, to make them easier to look at and also easier to print, actually, we usually invert the image to get the bands to be dark against a light background versus light against a dark background, because that makes it a little bit easier to see. So these students did the minus FNU, which is to say the uncut PCR product, treat with the enzyme plus FNU, and you can see very clearly a size in the shift in the size of the band. Same with the HAE3, minus H means to say the uncut PCR product, plus the enzyme means the product will cut if given the opportunity, and you can see a very definite shift in the size of the band. Now, this person is homozygous taster because there's no uncut molecule of either type after the digestion. Now, I want to point out something right here. We are expecting to see two bands in each of the digests, but those bands are very small, and they kind of run with the smudgy stuff at the bottom where the primers and things are. So in analyzing our data, we're just going to look at the larger bands and not worry about the smudge at the bottom. So we're simply going to analyze untreated product, treated, untreated, treated, and to see whether there's a shift in size. Okay, so here's another nice one, and this is a good gel because it has all the genotypes represented. So the first set of 4 lanes, and I have to say the students loaded these in different orders. So when you look at the data to analyze, you have to look carefully at the markings at the top of the lane to know what's supposed to be in that lane, because they are not the same in all cases. Okay, so the first set of 4 is one student sample, and the untreated product, the small product that's untreated, that's a 221 base pair, and then treated, which is treated with the enzyme, but there's no shift in size. So we're going to call that a non-taster allele because it's lacking the cut site. Same with the FNU type product. We've got the uncut, which is to say the molecule, the PCR product itself without digestion, and then we add the enzyme to it, and nothing happens. And that's what we expect for a little t allele because there's the restriction site has been destroyed. So this person we're going to call little t, and we predict that they will not be able to taste the PTC paper. The next set of lanes is a homozygous taster person. You'll notice that these students put different, they loaded their samples in different order. Okay, the first lane is the one with the enzyme in it. It was 221 base pairs, but if you compare it to the one next to it, the minus enzyme, there is a definite shift in size with the enzyme versus the uncut product. Similarly, the next set of lanes, we've got one with the enzyme, one without the enzyme. The one without the enzyme is 303 base pairs, and the one with the enzyme shifted down, and it's now 238 base pairs. And there's no sign of an uncut product except for those sort of shadowy bands up at the top. So we call this person big T, because both of their alleles appears to cut with the enzyme. The next set is a heterozygote. And so this is going to show us what happens when you've got one little t allele and one big T allele in the same genome. And again, we've got the plus H and the minus. So if you look at the minus, that's the 221 base pair product. And when it's treated with the enzyme, you get, again, a 221 base pair product. But you also get a smaller one, which is 177 base pairs, indicating that the site was present and cut. Now, ideally, you'd have exactly half of your molecules cutting, and this is obviously a little bit less than half. But the existence of a significant amount of uncut as well as a significant amount of cut implies that the person is big T, little t. So one of their alleles for mom or dad does not cut, it's big T, and the other allele does cut. Similarly with the F, the 303 base pair product is here, and then you treat with the enzyme, and some of the molecules cut to give you the shorter product, and that's the 238 base pairs. But a significant amount of the molecule is not cut, implying that that does not have a site, and so that is what you would see for a heterozygous person. One of their alleles is big T, one of their alleles is big T, and the other allele is little t. Okay, the last set of lanes is a little more confusing, and data are never perfect in real life. If you look at the A3, you see the uncut product size, and then you see, with the enzyme, some cut and some uncut. So just from that information, the person looks to be heterozygous, where some of their DNA contains a site and some of it does not contain the site. But then you look at the F, and you can see the uncut product, and then you see that with the enzyme, it looks like it's all cut. There's no remaining uncut product in that digest. So there's ways that that can happen. The most likely explanation is that the person really is big T, except their second allele didn't cut for the A3 for some reason. Because it is generally assumed, although not necessarily always the case, that if you have one of the SNPs, one nucleotide, you have all three of them. So this could be a person that has one of the sites and not the other, because they don't obey the PAV versus AVI genotypes with the exact same nucleotide changes. But to keep things simple, I would call this a big T person where the A3 just didn't cut very well. Okay, so now we're going to move on to look at the actual handout samples that you need to fill out for your lab report. So you should follow along on the worksheet and start filling in the blanks. So what you're asked to do is look at the 25 sets of samples from students and assign genotypes for the of big T, little t to each of them, and write down the band sizes that they produced from each of the enzymes that we studied. So make sure you look at the samples, look at the loading samples to make sure that the lane markings are right for each of them. Okay, so this is from the handout. We have students number 1, 2, 3, and 4, and we're going to look just at student number 1 first. So notice that they loaded their samples, the 2 uncut PCR products on the 2 left lanes. So the minus FNU means the first PCR product without enzyme. Minus A3 means the second one without enzyme. So those two bands are 303 and 221 respectively. Okay, then we treated the 303 base pair product with the FNU, and you can see there's a lot of the uncut product still there, implying that that's a little t allele, and then we do have some of the cut product, implying that that's a big T allele. And this is consistent with what you see with the A3. The uncut product is 221, and then after treatment with the enzyme, you still have some 221, which is the non-taster. But then you also have the cut product, implying that it's a taster. So this person is heterozygous. They're big T, little t, because they have one of each of the alleles in their DNA. Okay, so reminder again, you need to be careful what is actually loaded into each lane. Look carefully at those. Also notice that you cannot see the smaller restriction fragments. What you're looking for is a shift in size of the initial PCR product to a shorter molecular weight, implying that the site was present and the enzyme cut, and so the PCR product is shorter as a result of the enzyme chopping off the end, basically. All right, so summary here. Student 1 is a heterozygote, big T, little t, one allele cut, the other did not. write these bands on the handout table. All right. So student 2 is also a heterozygote, and I think I will let you analyze the rest. Do notice that there's some other bands around somewhere, like student 3 has some very faint shadowy bands that are above the size of the regular PCR product, and then you have some smudgy bands near the bottom. Just ignore those, look at the major bands, and then analyze the genotype on that basis. So person number 3 is little t, both products are uncut, etc. All right, so this just describes the A3 a little more carefully than I did before. So again, student 1 is a heterozygote. I want to point out, real-life gels are rarely perfect. Sometimes one enzyme cuts and not the other, just like I showed you. And sometimes you get some weird stuff. Just ignore the weird stuff. Okay, for example, here's a gel, and it's got some nice PCR products. Notice also that when we're showing these for the students, we're not showing the whole gel. We're just chopping out the part of the gel which has the bands we want to look at. But notice there are some higher molecular weight bands that are there and some shadowy bands. And those are called PCR products, meaning we have no idea where those came from. But that's okay. You'll do fine. Do the best you can.